Hey biology, I am here to make a big review video about everything we know about DNA, what it's made of, and what we use it for. So first of all, I want to label some things here. We see these X's and they're huge chunks of DNA called chromosomes. They're found in the nucleus, so this big purple thing inside of the cell must be the nucleus of the cell. Their job is to be the instructions for life, and life is made of proteins. So these are the instructions for making proteins. If proteins are food, DNA is the cookbook. Once we zoom in as far as we possibly can on the DNA, we see some things we're familiar with. We see the sugar phosphate backbone in a double helix shape. And in between, we see nitrogen bases that are paired according to our favorite rule, where we look at George Clooney. And what that actually means to us is that adenine pairs with thymine, and guanine pairs with cytosine. We're next going to compare DNA and RNA because there's more than one type of nucleic acids. These are both nucleic acids, and we know that because they have the elements P, C, H, O, and N. There are some more things we know about the differences between the two. We know on the left we have DNA because we see a double helix, number one. Number two, we see that we have adenine, thymine, guanine, and cytosine. So there's something special that DNA has that RNA doesn't. It has thymine. Our last thing that makes DNA unique is that its sugar is called deoxyribose. So I just picked a random sugar and I'm going to label it deoxyribose. So if I asked you for the three things that make DNA unique, what would you say? You would say it's a double helix, it contains thymine, and the sugar inside is deoxyribose. Next, let's talk about RNA. I'm going to change colors to red. So it has all the same elements inside, but the key differences are you see only a single strand. It's still twisted, but there's only one strand, and the nitrogen bases kind of stick out from it. Here's our next difference. We have uracil, guanine, adenine, and cytosine. Which one of those is not found in DNA? You're right if you said uracil. That's unique to RNA. Last one, in our sugar phosphate backbone in purple, you're going to find phosphate groups and a sugar called ribose. So if I asked you for the three unique qualities of RNA, what would you say? You would think it's single-stranded, contains the nitrogen base uracil, and the sugar ribose. So we have two different sugars, two different shapes, and we switch thymine out for uracil. There are really important similarities too. Let's not forget that all the elements are the same, and let's not forget that they all have phosphates inside of them, phosphate groups. And that's in the backbone of RNA and the backbone of DNA. We're moving on to a type of replication that's called semi-conservative replication. Let's break down that word. Semi means half, and conservative means old, keeping older things. So when we replicate DNA, we keep half of the old double helix. Here's what we mean when we say that. This is our original double helix, and it is being unzipped or split apart. What's the enzyme that unzips DNA? You remembered right if you said that right here we should have the enzyme called helicase, unzipping the DNA. After that, our old template strands have a special job to do. They sit single-stranded and wait for new nucleotides to be attached to them. The most important thing happening here is, again, our base pairing rules, where A goes with T and C goes with G. So if your old template strand has an adenine on it, your new strand is going to have thymine. And we're going to follow that rule all the way down. So at the end, we have 2 new double helixes, or helices, and they're each half old and half new. That top one's half old, half new, and then the bottom one is half old, half new. What we're moving on to next is the job of DNA. So DNA isn't there to just be there, it's there to make proteins. And proteins make you who you are. So making protein is a 2-step process, and I'm going to teach you how your body accomplishes both steps. Basically what happens is DNA is like a princess locked in a tower. It's trapped in the nucleus because it has to stay safe. If something bad happened to your DNA, that would be called a mutation, and that would be really, really bad news for you. So if DNA changes, if one of the nitrogen bases changes, that's called a mutation. And we have to watch out for those. So that's why DNA stays in the nucleus. Because it's in the nucleus, we need to write down what it has to say so that we can leave with that copy. So imagine DNA is the princess locked in the tower. A knight comes to the tower and says, "Princess, what would you like?" And the princess says, "I would like to order new curtains." That's what she wants. She wants curtains. Transcription is when we write down the message of DNA. It's when we copy what DNA has to say. That copy is called messenger RNA. It's not a protein yet, so we need to go through one more step. After we copy down what DNA has to say, we have to transform it and make it into a chain of amino acids. This makes sense that it's called translation because we're going from the language of nucleic acids to the language of proteins that are made of amino acids. In transcription, you go from DNA to RNA, and in translation, you totally change languages. You go from RNA to protein. Just like if Mr. Garcia asked you to translate something, you would take it all the way from English to Spanish. Let's look at some details. Here we have DNA. It's being opened up or unzipped by helicase. And we are making messenger RNA based on what the DNA has to say. So what process is this? It is transcription. Where is this happening? Well, remember your DNA is locked up in the tower. So this is happening inside of the nucleus. How is the messenger RNA made? We use the base pairing rules, but they're a little different because we have RNA instead of DNA. So paired to adenine, we have uracil. Paired to guanine, we have cytosine. Paired to thymine, we have adenine. And the most important part, remember, paired to adenine, we have uracil because this is messenger RNA. Why should we do this? We need to get the message of DNA. We need to get DNA's recipe all the way to the protein factory of the cell. We call those parts of the cell the ribosome. The issue here is DNA can't leave the nucleus. So to get DNA to the protein factory, we need to copy down what it has to say. And another word for copying down is transcription. All right, now that we've gone through all of transcription, let's take this transcript and leave the nucleus. Our next step is we take our messenger RNA and we totally change the language. We are going to translate RNA into protein. Here's how it happens. It's like a secret decoder ring. We call these triplets of 3 nitrogen bases a codon. So here's one messenger RNA codon that says UGA. We transform one codon into one amino acid. Here's how it happens. We have these little goofy things that look like sorry pieces. They're called transfer RNA. Their passenger is the amino acid they bring, but how do they know where to drop him off? They know where to drop him off because they have something on them called an anticodon. And it's complementary to the codon. So we'll match A with U, G with C, and U with A. Remember, here's our anticodon on tRNA, and here's our codon on messenger RNA. How many nitrogen bases are in a codon? There are 3. 1, 2, 3 nitrogen bases. How many amino acids does that translate into? Just 1 amino acid. This is a big picture, literally, of everything we've already learned. We have transcription as step 1. It has to happen first because DNA is trapped in the nucleus and we need to get it out. So we make our messenger RNA and it leaves the nucleus. Where does it go? It goes to the ribosome, which is the protein factory, and it goes through step number 2, which is translation. What happens in translation is messenger RNA codons are matched up with their transfer RNA anticodons. This has to happen so that transfer RNAs can drop off the right amino acid to the right location. What I want you to take away from this is how careful we have to be in this process. So, when we're doing transcription, we need to be very careful that we always match things up correctly. If we made a mistake in transcription and we ended up with an incorrect nitrogen base, that means that when we make it to the ribosome, we are going to have the wrong codon. If we have the wrong codon, it's like you giving the taxi driver the wrong address. tRNA is going to drop off the wrong amino acid. And your protein might change in a way that might hurt your body. That is it for the pictures. We're going to go through 2 animations together. We're about to do 2 different simulations together. The first one is going to show us replication, and then the next one will go through transcription and translation. So when we're going through replication, the first thing we need to do is take an original DNA double helix and unzip it with helicase. After it's unzipped, we have 2 old template strands, and we need to add new nucleotides to it. We decide which ones to add using the base pairing rules. So T pairs with A, G pairs with C, and we keep going from there. Cytosine to guanine, adenine to thymine. What I want you guys to notice is I'm doing this like double time. So I'm building a new strand on the left and a new strand on the right. That makes sense because my original DNA split in half. So cytosine pairs with guanine, adenine to thymine. You guys do the next one. Adenine pairs with, you got it if you said thymine. And my guanine will pair with, you got it if you said cytosine. I have another simulation for us to finish up with. This one is going to have us transcribe a gene and then translate a gene. So here's our DNA strand. The colored boxes represent our nitrogen bases. And we're going to transcribe it. We're making messenger RNA in the nucleus. A pairs with, don't say T, because we are making RNA. A pairs with U. T pairs with A. Next we'll have adenine, and next we'll have uracil. guanine, cytosine, and I'll just do the rest. All right, we made our messenger RNA strand and it left the nucleus. The next thing we need to do is transform it into a protein. So here's how we do it. We want to find the start codon, and it's a triplet, so it's 3 nitrogen bases that give us the amino acid methionine. If you look at our genetic code, here's methionine, it's the start codon. It is AUG. Here's how you would find it. You go to that big red A, the yellow U at the top, and then you find G in green at the right, and here it is, methionine. So let's mouse over our start codon. Is it here? Nope. How about now? Nope. There it is, AUG. That's where we'll start translating. Don't start too early. Now we're going to translate by dragging the right amino acid over to our messenger RNA. We are making a protein when we chain amino acids together. So we'll start with methionine. Next we have CUA. Purple C, yellow U, red A. CUA is leucine. Now that we have 2 amino acids bonded together, we've just started making our protein. Next, try it yourself. Find us the amino acid for the codon GAC. GAC is aspartic acid. Hey, aspartic acid, come here. There it goes. Next we have GUG, which gives us valine, UUC, which gives you phenylalanine, and UAG. Hey, it's time to stop. Fantastic. We created our own protein. It started with a start codon, it stopped with a stop codon, and everything looks great. It's important that we always have the correct codons, because if that gets messed up, our protein gets messed up, and that's what's running your body, so you better watch out.